Glyoxylate cycle. Glyoxylate cycle discovered by Beavers and Kornberg scientists. Beavers and Kornberg scientists discovered glyoxylate cycle. Beavers and Kornberg shown that maltase synthase and isocitrate lyase. These two enzymes are important for the glyoxylate cycle. One is maltase synthase and second one is isocitrate lyase. The two enzymes are important for glyoxylate cycle. The two enzymes that characterize the glyoxylate cycle were present in the endosperm of castor beans. In plants, the glyoxylate cycle occurs in special peroxisomes, which are called glyoxysomes. Peroxisomes simply we called in plants glyoxysomes. This cycle allows seed to use lipid as a source of energy to form the shoot during the germination. The seed cannot produce biomass using photosynthesis because of lack of an organ to perform this function. Means seed need compulsory energy. That energy is collected from the or used from the lipids. Seeds didn't have any chloroplast, so the seeds cannot participate in the photosynthesis. So photosynthesis is completely absent in the seeds. So at that time the seed need energy. That energy is gained from the stored parts. The stored parts are called lipids. The energy is collected from the used from the lipids for the germination purpose. The glyoxylate cycle particularly important in species which synthesize carbohydrates from two carbon substrates such as ethanol or acetate, and in germinating plant seeds which must synthesize their carbohydrates from stored triacyl glycerols. These are the lipid molecules. Triacyl glycerols are the stored lipid molecules. From this stored lipids, from this stored triacyl glycerols, the plant seeds gain energy. The glyoxylate cycle, a variation of the tricarboxylic acid cycle, is an anabolic pathway occurring in plants, bacteria, protists, and fungi. The glyoxylate cycle centers on the conversion of acetyl coenzyme A to succinate for the synthesis of carbohydrates. Generally, tricarboxylic acid pathway or tricarboxylic cycle present in the or occurs in the mitochondria. Similar, the tricarboxylic pathway cycle reactions are occurs in the glyoxysomes. That glyoxysomes are produce special type of products by the glyoxylate cycle. CRT is the stored parts, stored food material like oil bodies in the cells. The oil body containing lipids. The lipids are triacyl glycerols. The triacyl glycerols are converted into fatty acids. The fatty acids are enter into glyoxysomes after activation. Fatty acids are activated. That activated fatty acids are enter into glyoxysomes. The activated fatty acids are called acyl coenzyme A. The acyl coenzyme A containing long chains of fatty acids. Long chains of fatty acids. From this long chain of fatty acids, activated fatty acids are acyl co-MgMA. Only two carbons are released as a acetyl co-MgMA. Acetyl co-MgMA. Remaining carbons are present as T's as a acyl co-MgMA. The activated acyl co-MgMA are having long chains of carbons. From these long chains of carbons, only two carbons released as a acetyl co-MgMA, and remaining carbon chains are present as it is in the form of acyl co-MgMA. Just we discuss one example here. It is this acyl co-MgMA containing 10 carbon atoms. From these 10 carbon atoms, 2 carbon atoms are released as a acetyl co-MgMA, and remaining 8 carbon atoms are always called acyl co-MgMA. By this process, 2 carbon atoms of acetyl co-MgMA is released. The acetyl co-MgMA is combined with 4 carbon atoms of oxaloacetic acid. 2 carbon atoms of acetyl co-MgMA combines with 4 carbon atoms of oxaloacetate to form citrate, 6 carbon atoms of citrate in presence of citrate synthase enzyme. Here this citrate synthase enzyme is encourage this reaction. The 6 carbon atoms of citrate convert into isocitrate in presence of aconitase enzyme. 6 carbon atoms of isocitrate, 6 carbon atoms of citrate convert into 6 carbon atoms of isocitrate in presence of aconitase enzyme. See, here it is up to these reactions are similar in the tricarboxylic acid cycle, like in Krebs cycle. The same reactions are occurs in the TCA cycle also, but the TCA cycle occurs in the mitochondria, not in the glyoxysomes. The similar reactions, the similar 1 or 2 reactions are occurs in the glyoxysomes in the glyoxylate cycle. See, the 6 carbon atoms of isocitrate is formed in glyoxysomes. It involves in the bypass reaction. It cleaves into 2 parts. One is 2 carbon atoms of glyoxylate and 4 carbon atoms of succinate. Isocitrate is cleaves into 2 parts. One is glyoxylate, it containing 2 carbon atoms and 4 carbon atoms of succinate. The glyoxylate containing 2 carbon atoms, succinate containing 4 carbon atoms. For this reaction, isocitrate lyase enzymes is involved for this reaction. It is important enzyme, first enzyme for the glyoxylate cycle. Isocitrate cleaves into 2 carbon atoms of glyoxylate and 4 carbon atoms of succinate. For this reaction, isocitrate lyase enzymes is helped for this. And 2 carbon atoms of glyoxylate combines with 2 carbon atoms of glyoxylate combines with 2 carbon atoms of acetyl co-MgMA in presence of maltase synthase enzyme to form maltase. These are the 2 enzymes for this glyoxylate cycle. What are the 2 enzymes here? It is one is isocitrate lyase and second one is maltase synthase. 6 carbon atoms of isocitrate cleaves into 2 parts. One is 2 carbon atoms of glyoxylate and 4 carbon atoms of succinate. So first product in this bypass reaction in the first product is glyoxylate. So this cycle is called glyoxylate cycle. For this reaction, important enzyme, bypass enzyme is isocitrate lyase. Then after this reaction, the two carbon atoms of glyoxylate combines with the two carbon atoms of acetyl coenzyme A to form four carbon atoms of malate in presence of malate synthase. It is the second enzyme, second important enzyme for the glyoxylate cycle. These two enzymes are play major role in the glyoxylate cycle. One is isocitrate lyase, second one is maltase synthase. Finally, after maltase synthase enzyme reaction, four carbon atoms of maltate is present. Maltate is formed. The maltate convert into four carbon atoms of oxaloacetate in presence of malate dehydrogenase. In this reaction, one NAD plus convert into NADH and four carbon atoms of oxaloacetate. Some oxaloacetate are combined with acetyl coenzyme A. They involved in the formation of citrate, six carbon atoms of citrate. Sometimes some oxaloacetates are enter into cytoplasm. They are involved in the gluconeogenesis. From these four carbon atoms of oxaloacetate, glucose is formed by the gluconeogenesis cycle. Here it is one pathway, and the four carbon atoms of succinate, enter into mitochondrial matrix. The four carbon atoms of succinate enter into mitochondria of TCA cycle, tricarboxylic acid pathway. The TCA cycle received four carbon atoms of succinate and it convert into four carbon atoms of malic acid. The four carbon atoms of malic acid convert into four carbon atoms of oxaloacetate. The four carbon atoms of oxaloacetate also enter into cytoplasm. And these four carbon atoms of oxaloacetate convert into three carbon atoms of one product, that is phosphoenol pyruvate, and one carbon released as a carbon dioxide. And these three carbon atoms of PEP, phosphoenol pyruvates, here it is two molecule, here is only one molecule is there, similar cycle occurs in this. Two phosphoenol pyruvates are involved in the gluconeogenesis cycle. Two PEP, two molecules of PEPs are involved in the gluconeogenesis cycle and they develop into glucose or sucrose. By this glyoxylate cycle, oxaloacetate is released, enter into the oxaloacetate, four carbon atoms of oxaloacetate enter into cytoplasm through the glyoxysomes. At the same time, the four carbon atoms of oxaloacetate enter into the cytosol from the mitochondria. These four carbon atoms of oxaloacetates are convert into glucose or sucrose. It gives energy to the seeds for the germination. The glucose or sucrose gives energy for the seed germination. So by this process, seeds gain energy for the germination of seeds. It is a glyoxylate cycle.